Hello learners, this is Habeeba with Excelwithme. If you like the video, please do like, share, subscribe and comment below. We were talking about the research methodology paper, tests of significance topic. Under that concept, utility and computation of standard error. In the previous class, we have understood the meaning of standard error. Now we are going to understand tests of significance here. In statistics, tests of significance are the method of reaching a conclusion to reject or support the claims based on sample data. When we take any problem, we give some objectives and then we decide the assumptions, that is tentative solutions of the problem, that is hypothesis, and that hypothesis will be tested through some statistics after collecting the observations. So to come to the conclusions, we need some statistics and also test of significance, whether whatever hypothesis we have done, whether it should be accepted or rejected. The statistics are a special branch of mathematics which deals with the collection and calculation over numerical data. This subject is well known for research based on statistical surveys. During a statistical process, a very common as well as an important term we come across is significance. That is statistical significance. Now what is it? It is very important in research, not only in mathematics but in several different fields such as medicine, psychology and biology, and also education. There are many methods through which the significance can be tested. These are known as significance tests. Let us learn about significance testing in detail now. Now, what is the definition of significance testing? In statistics, it is important to know if the result of an experiment is significant enough or not. Whatever research problem we have taken, whether the results are significant enough or not, how we will understand through these tests. In order to measure the significance, there are some predefined tests which could be applied. These tests are called the tests of significance or simply the significance tests. This statistical testing is subjected to some degree of error. For some experiments, the researcher is required to define the probability of sampling error in advance. In any test which does not consider the entire population, the sampling error does exist. So, the testing of significance is very important in statistical research. Now, the significance level is the level at which it can be accepted if a given event is statistically significant. This is also termed as p-value. Very important term we have to remember for the statistics, p-value. It is probability value. It is observed that the bigger samples are less prone to chance, thus the sample size plays a vital role in measuring the statistical significance. One should use only representative and random samples for significance tests. Now, tests of significance, confidence intervals are one of the two most common types of statistical inference. Researchers use a confidence interval when their goal is to estimate a population parameter. The second common type of inference, called a test of significance, has a different goal. What is that? To assess the evidence provided by data about some claim concerning a population. Now, let's see the concept of test of significance here. A test of significance is a formal procedure for comparing observed data with a claim, also called a hypothesis, the truth of which is being assessed. Whatever hypothesis, that is tentative solution we have given in our below the objectives, we have to test whether it is true or false. The claim is a statement about a parameter like the population proportion P or the population mean, that is mu. The results of a significance test are expressed in terms of a probability that measures how well the data and the claim agree. Now, what is the utility of this test of significance? Let's understand. The significance is the probability that a relationship exists. That is, significance tests tell us about the probability that if a relationship we found is due to random chance or not, and to which level, that we will understand. This indicates about the error that would be made by us if the found relationship is assumed to exist. Now, tests of significance in statistics. Technically speaking, the statistical significance refers to the probability of a result of some statistical test or research occurring by chance. The main purpose of performing statistical research is basically to find the truth. That is, in this process, the researcher has to make sure about the quality of sample, accuracy, and good measures which need a number of steps to be done. Now, the researcher has to determine whether the findings of experiments have occurred due to a good study or just by fluke. Now, the significance in a number which represents probability indicating the result of some study has occurred purely by chance. The statistical significance may be weak or strong. It does not necessarily indicate practical significance. Sometimes when a researcher does not carefully make use of language in the report of their experiment, the significance may be misinterpreted. Now, the psychologists and statisticians look for a 5% probability or less, which means 5% results occur due to chance. The rest of them will be true. This also indicates that there is a 95% chance of results occurring not by chance. They are by the hard work, the evidence is collected, statistics done, that is taken into consideration. Whenever it is found that the result of our experiment is statistically significant, it refers that we should be 95% sure the results are not due to chance. Now, what is the process of significance testing? In the process of testing for statistical significance, there are the following steps. Number 1, stating a hypothesis for research, stating a null hypothesis, selecting a probability of error level, then selecting and computing a statistical significance test, lastly interpreting the results. Now, what are the types of errors? There are basically two types of errors, type 1 and then type 2. Type 1 error, what is type 1 error? The type 1 error occurs when the researcher finds out that the relationship assumed through research hypothesis does exist. That is, but in reality there is evidence that it does not exist. In this type of error, the researcher is supposed to reject the research hypothesis and accept the null hypothesis. But its opposite happens. The probability that researchers commit type 1 error is denoted by alpha. So what happens in type 1 error? We reject when it is true. Whatever hypothesis when we have done, when it is true, we are rejecting. That means that is type 1 error. Now, type 2 error is just opposite the type 1 error. It occurs when it is assumed that a relationship does not exist. That is, when it is false, we are accepting. But in reality, it does not. In this type of error, the researcher is supposed to accept the research hypothesis and reject the null hypothesis. But he does not, and the opposite happens. The probability that a type 2 error is committed is represented by beta. Now, types of statistical tests here, one-tailed and two-tailed are two types of statistical tests that are used alternatively for the computation of the statistical significance of some parameter in a given set of data. These are also termed as one-sided and two-sided tests. In research, the one-tailed test can be used when the deviations of the estimated parameter in one direction from an assumed benchmark value are considered theoretically possible. On the other hand, the two-tailed test should be utilized when the deviations in both directions of benchmark value are considered as theoretically possible. When we take the graph here, these are the two-sided. When we take, it will slope like this and it will go to one side. This is one-tailed, and if we are taking the two sides, that will become the two-tailed. The word tail is used in the names on these tests since the extreme points of the distributions in which observation tend to reject the null hypothesis are quite small and tail off to 0. Similar to the bell curve or normal distribution just now I have shown, the choice of one-tailed or two-tailed significance test depends upon the research hypothesis. Now example, the one-tailed test can be utilized for the test of the null hypothesis, such as boys will not score significantly higher marks than girls in 10th standard. In the 10th standard. In this example, the null hypothesis does not indirectly assume the direction of the difference here. Now the two-tailed test could be utilized in the testing of the null hypothesis. There is no significant difference in scores of boys and girls in 10th standard. Now what is p-value testing? In the context of statistical significance of the data, the p-value is an important terminology for hypothesis testing. That is, the p-value is said to be a function of observed sample results which is being used for testing of statistical hypothesis. A threshold value is to be selected before the test is performed. This value is known as the significance level, that is traditionally 1% or 5%. It is denoted by alpha. In the case when the p-value is smaller than or equal to significance level alpha, the data is said to be inconsistent for our assumption of the null hypothesis to be true. Therefore, the null hypothesis should be rejected and an alternative hypothesis is supposed to be accepted or assumed as true. Note that the smaller the p-value is, the bigger the significance should be, as it indicates that the research hypothesis does not adequately explain the observation. If the p-value is calculated accurately, then such test controls type 1 error rate not be greater than the significance level alpha. The use of p-values in statistical hypothesis testing is very commonly seen in a wide variety of areas such as psychology, sociology, science, economics, social science, biology, criminal justice, etc. Now, what is standard error formula, which we have seen in the previous class? Accuracy of a sample that describes a population is identified through the standard error formula, the simple mean which deviates from the given population, and that deviation is given as SE mean equal to S, that is standard deviation, divided by square root of n, where S is the standard deviation and n is the number of observations. Now standard error of the mean in the previous class we have understood. You can pause it and revise this again. SEM equal to S divided by square root of n. Now standard error of estimate, that is SEE. The standard error of the estimate is the estimation of the accuracy of any prediction. It denotes as SEE. Formula is given where Xi stands for data values, X bar is the mean value and n is the sample size. Now how we compute this standard error, very important here. Step 1, note the number of measurements, that is n, how many observations, and determine the sample mean mu, it is average of all the measurements. Then step 2, determine how much each measurement varies from the mean. Step 3, square all the deviations determined in step 2 and add together, that is standard into mean I minus mu whole square. Step 4, divide the sum from step 3 by 1 less than the total number of measurement. Here you can pause it and see all the steps given here. Now let's see the example here, standard error example. Calculate the standard error of the given data y equal to 5, 10, 12, 15, 20. That is how many observations? 1, 2, 3, 4 and 5 observation, n equal to 5. Solution first, we have to find the mean of the given data. Mean equal to, we have to add all the number divided by 5, that is 10.5 is our mean. Now the standard deviation can be calculated as S equal to summation of difference between each value of given data and the mean value or number of values. Then hence you can see the calculation here. After solving the above equation we get S equal to 5.35. Therefore SE can be estimated with the formula SE equal to S divided by square root of n. That is, you can get the answer 2.39. Standard error is 2.39. Now standard error versus standard deviation. Let's see here. The below table shows how we can calculate the standard deviation using population parameters and standard error using sample parameters. You can pause the video here and you can check it out. The population parameters, mean, sample proportion, difference between means, difference between proportions P1 minus P2, and then formulas are given in the second column, and the third is the sample statistics, and fourth column is the formula for SE, standard error. Thanks for your attention and time. If you like the video, please do like, share and subscribe and comment below. Thanks for your attention and time. Happy learning.